Our presentation is on prenatal DNA sequencing. Prenatal DNA sequencing is important for several reasons. First, it allows doctors to diagnose potential diseases earlier in pregnancy. This can allow doctors to make better choices for the safety of the mother and the child, such as understanding whether a child is predisposed to diabetes or some other disease. The emerging technology that we are going to discuss is much safer than current methods. However, DNA sequencing in general are very expensive, costing $10,000 for an adult and $50,000 for a fetus. This process can be made cheaper by doing partial sequencing. Current methods of prenatal DNA sequencing introduce an increased risk of miscarriage and are invasive because they require taking fetal cells from the placenta or amniotic fluid of the mother. DNA sequencing does carry some ethical issues, considering 65% of women choose abortion when they find out that they are going to have a child with Down syndrome. There's also an issue that people may put too much faith in the genetic information, such as assuming their child will have diabetes if there's a predisposition for it.
This is the technique for sequencing the DNA gathered from the blood of the mothers whose babies are being tested for different diseases. The first step of the technique is to take the DNA that you have gathered from the blood of the mother, which in this case is the green dots, so that's the DNA that you're going to want to be working for and learn the sequence of. You take that DNA and you break it into smaller fragments, which then you can more easily analyze. And after that, you have to add adapters to the end of the fragments so that throughout this technique, the DNA can be copied and be amplified into more of the same sequence of DNA. Step 2 begins with the attaching of one of the DNA strands from the DNA fragment that you have to a chip or a plate that has the complementary adapter sequences on it. This allows the fragments to bend into an upside down U shape by being attracted to the complementary adapter sequences and lightly bonding to them. From there, you can add DNA bases to the chip or the plate, which then create a sequence of DNA that is complementary to the one that you have. This allows you to, when you increase the temperature, break the bonds, and you now have two of the original strands. However, one is the complementary strand. This would be a problem unless you repeated the process and caused the complementary strand to be complemented back into the original strand of DNA. And by doing this, you create a cluster of that original fragment in the local area on the chip. You can then wash off the complementary strand, which you don't need. This is a picture of step 2. Start with the single strand of DNA that you want. It bends over and matches the complementary sequence on the chip, and then goes and makes the complementary DNA fragment, which then can go back up into single strands, bend down again in another spot, make another complementary strand, straighten up, and do this a bunch of times, and then you have a single strand fragment that you want for the sequencing part. On the last picture, there is a extra adapter sequence at the top for initiating the next step, which is sequencing from there. The third step of the technique is to take the adapter sequence that we saw at the top of the strand on the last slide and add a DNA base to it. This base has been marked with a specific color that, under certain conditions, fluoresces, so that you can see that color, and that color is specific to that base. So an A would be a certain color, a T would have a color, a G would have a color, and C would have a color. Um, then you make the base fluoresce and you take a picture of it. You add another DNA base to the strand and then take a picture of that once it's fluoresced. And then you repeat this process until you have the entire DNA strand in color form with a bunch of pictures taken after you've added each base. This is a diagram for step 3. The adapter sequence is at the top of the strand in blue, and followed by the first base, which is in this case a T, and it fluoresces blue. On the far right, the picture that's taken at the top of that cluster is blue, and so you would know that that first base is a T. This continues. The first one that came out was a T, followed by a G, followed by a C, followed by a T, followed by an A, followed by a C, which then means that the original strand is the opposite: A, C, G, A, T, G. Almost all of this is done by a computer or in a machine that you can buy. Well, but a company can buy them.
We can see that this emerging gene sequencing technique is much safer because it requires a sample of the mother's blood and not some more invasive procedure. It will also allow doctors to make better decisions, and costs will become feasible as the technology matures. Still, there are ethical questions that must be considered. UAG. See? Get it? It's a stop codon, right? Okay. Never mind, you don't guess. The end.